Hello and welcome to Food Safety Fridays. My name is Simon Simpley from the International Food Safety and Quality Network. We're back. Say we're back, Maria.
We're back. Welcome back.
And what a show we've got for you today. Maria Sandoval from Trace Analytics. And today we're going to be talking about indicator organisms and food grade air. How are you doing, Maria?
I'm doing well, Simon. I'm usually in Austin, Texas, but today I'm in Ocala, Florida. So I had to run away into a conference room to come do this webinar this morning.
Well, we really appreciate you doing that, pulling you away from that. But it's great to have you. We've not actually, it's almost a month since we did the last webinar. So
yeah,
back onto it now. Tell us where you're joining from in the sidebar. It's great to see you all, new people and repeats, regulars. Lovely to see you. Tell us where you're joining from. Play the video ads now, and we'll be back in a couple of minutes for presentation. In a second, sorry. There we go. Okay, I hope you could see those, the video okay then. That's kind sponsors of IFSQN, and one of those is Trace Analytics, been a sponsor for a long time, great friends of ours, and Maria Sandoval, fantastic speaker. I'll be back for the Q&A later. For now, I'll hand you over to Maria.
Hi guys, good morning. This is Maria Sandoval. I'm going to talk about indicator organisms in food grade compressed air. So a little bit about the company I work for, Trace Analytics. We've been in compressed air testing since 1989. We are 17025 accredited. And what does that mean? And why is that important to a food and beverage company? Well, to have the most compliant and the most quality assurance in your testing lab, you want to make sure that you pick a third-party lab that's accredited. And that is something that Trace Analytics can do. We test to ISO 8573 for compressed air. We test to ISO 14698. Also for compressed air, ambient air, environmental samples, surface samples, and we test to many other food and beverage standards. I currently sit on the NFPA 1989 board along with some other members, and we have state-of-the-art equipment here at Trace, and we have clients all around the world. So don't hesitate to ask if you're interested in anything regarding needing help. A little bit about myself. I'm the lab director here at Trace. I have over 20 years of experience in microbiology and molecular biology. Before I entered the world of compressed air, I was in molecular carcinogenesis doing cancer research. Now I've pivoted over to the microbiological world in compressed air, and I have some amazing things to talk to you guys about today. I sit on the ISO 8573 committee and am currently the project leader for 8573-7, which is in particular the microbiological contamination section. And it talks about how you sample for microbiological organisms in compressed air. So we're in the process of rewriting that, but we will also be hosting the ISO 8573 committee in Austin, Texas in September. So if you're on the ISO board or you want to become an expert, please, we would love to have your input from food and beverage standpoint. I'm also on the ISBT Gas Technical Committee, so you have any questions regarding that, and FBA 1999 and ISPE. So let's go ahead and get started. One of the questions you need to be asking in food and beverage is, why do you care about the quality in compressed air and gases? And particularly when you look up quality and you look up compressed air together, you'll find this ongoing theme, and it's basically discussing how compressed air is an industrial utility. It's considered the fourth utility in the world. It's something that is used every single day. So when you're thinking about your food and beverage facility, think about how many pneumatic tools you use. Think about if you're using compressed air as an ingredient. Think about if you're using compressed air just as indirect contact. So these are all the types of questions and the reasons why we care about the quality of compressed air and gases. And so when you're thinking about, well, Maria, why is it so important? I don't understand. We care about controlling the environment. Compressed air is just a tool that we use. Right. Well, 1 cubic meter of untreated compressed air contains about 200 million dirt particles, water, microbes, and oil. And one of the most important things to remember is that ambient air has all of these particles in it, right? That's not the concern. The concern is when you take in ambient air and you compress it into a storage tank, you're also concentrating all of those contaminants per cubic meter. And so the contaminants that you want to worry about when you're thinking about compressed air in food and beverage are particles, water, oil, microorganisms, contaminant gases. Today, we're going to focus on microbial contamination, kind of what that looks like and how to use indicator organisms as a way to facilitate the health and safety of your compressor system. So we talked a little bit about where these microbial contamination comes from, but it's really coming from the ambient environment. And once it gets sucked into your system, it is solely dependent on the health of your compressor to make sure that you guys are eliminating any possibility to move those microorganisms that get taken in from the intake all the way to your point of use. And so you're looking at filtrations, you're looking at dryers, piping systems, things like that. And so you want to ask yourself what you expect of your point of use. Do you expect that the compressed air will be used as an ingredient? Do you expect that the compressed air will be used strictly as a tool? It'll be loaded for a pneumatic tool. And so once you make that decision, and so let's say that when we're going into food grade compressed air, you have decided that this compressed air is going to be an ingredient. And so now from a quality standpoint, you need to find a standard that'll guide you through your risk assessment to figure out how you're going to start testing. And so there are a couple bodies that drive the food safety standards and regulations. And that's ISO. ISO is a standards body, and they create methods for testing compressed air contaminants. The FDA has regulations in the United States, which is adopting SQF as well. And so SQF is an international standard that utilizes the technology that is brought forward from the ISO standards, and BRC Global Standards also references the ISO methods. And so the reason that you want to pick a standard is because if you have too much diversity, it creates complexity. So if you have too many people telling you how to do something and not a unified body, you create too many different ways to accomplish the same goal. And so what we're trying to do is compare apples to apples and oranges to oranges when it comes to food grade compressed air. You want to avoid the confusion that comes with that complexity. And to prevent that, you need that commonality, which is ISO. And so standardization brings understanding and unity across the board and creates a uniform version of really food grade compressed air. So some of these standards talk about microorganisms and some just allude to microorganisms. So for ISO 8573, the test methods and purity classes for compressed air, many of you probably have compressed air that is 222 or 232. Those particulate purity classes do not differentiate between viable and non-viable particulates. So what does that mean? That means that particulate contamination is being encumbered with microorganisms because microorganisms are a particle, and they are not, in your purity classes for particulate, are not differentiating those two. We also have another ISO standard that talks about microorganisms in controlled environments, specifically in your compressed air. So ISO 14698 is a standard that talks about biocontamination in controlled environments. So for many of you who are doing ready to eat foods, or if you have a clean room in your food facility and you use compressed air, ISO 14698 is a great tool, but it doesn't tell you how to do the testing. So when you're going and you're saying you want to be compliant to ISO 14698, you would lean on ISO 8573-7 for those test methods. But since there's no clear guidance on what microorganisms to look for in food grade compressed air, we're left in this kind of this ether of possibilities. And so that isn't something that's true for end product food and beverage. For a lot of you guys, you are going to be familiar with the terminology indicator organisms from water. And so we need to figure out why we can't marry these two worlds. Air is air, whether it's compressed or it's ambient. They still have the ability to carry these contaminants. So, I would challenge you to start thinking about why, we're cherry picking ambient air over compressed air. So when you're thinking about indicator organisms, one of the things I was thinking about while I was creating this presentation was, where have I heard that before? How am I going to tie in indicator organisms to compressed air testing? Because typically when you hear the terminology indicator organisms, and there's always one microbiologist on this call, so I am clearing this up, it is often referred to for water quality testing. It's a way to say, hey, our water is probably we have an indicator organism positive. The water is contaminated somewhere by waste. But I would like to challenge, the industry to think of indicator organisms also as a way to signify an indication that our compressor system is having issues. And that's by saying if we have the presence of bacterial or fungi species in our compressor, there is an indication that something somewhere along the piping system has broken down. And that doesn't mean physically broken down, but it means we've lost control somewhere. And it really begins with understanding your system. So some of the indicator organisms that are available and that are typical are, let's say, food and beverage, and you deal with let's say you deal with lettuces, or you deal with vegetables, and you're concerned about listeria and you're concerned about E. coli. Well, indicator organisms mean that you have an understanding of the characteristics. So we know that E. coli and listeria are gram negative and gram positive. We know that they are rod shaped. We know that they also have severe health and safety concerns. It can cause end product damage. It can cause brand reputation issues. It can cause health and safety issues, not only to your employees, but to the end users themselves. And so these are the types of organisms: Listeria, E. coli, coliforms, Salmonella, Shigella, Candida, that are available to be tested as a way to indicate whether A, you have severe pathogens in your compressed air, or B, whether you have a breakdown somewhere in your compressor system. And there's a couple of ways to look at it that way. Let's see. And so when it's time to test for those indicator organisms, you need to be using an impaction sampler. And so what you see here on the screen is, what you need to see on the screen is the point of use is connected through a hose. And that is going to drive the compressed air through our system into these valves down here and allow the compressed air to impact an agarose plate. And that will be sent to a third-party laboratory or your in-house laboratory to satiate the requirements of ISO 8573-7 or 14698. And so both of these standards require the use of an impaction sampler. And so here at Trace, we provide these as well. We do them as rentals, or you can get them as a purchase. It's also not a requirement to use, just any piece of equipment. You need to be able to quantify the CFUs. There are many options for samplers, but not all samplers are equal. So make sure you do your homework when you're doing equipment choices. And so the second thing that's important when you're doing sampling for indicator organisms in compressed air is making sure that your technician or whomever is doing the analysis is taking aseptic technique to the next level. So they have a general understanding of aseptic technique. And I think during COVID times, we all learned how to wash our hands a lot. We learned how to use 70% alcohol spray and wipes. But this is imperative to doing a high quality test. And the added benefit is that it's not hard. So one thing that people constantly tell our laboratory here at Trace is, especially if they're first-time users, is I'm scared. I don't know if my technician's going to be able to do this. This seems like a scientific thing. It seems like I need somebody to who is, super scientist to do this. But the truth is, you just need foundational knowledge of aseptic technique. And here at Trace, we provide an academy that your technicians can go through to learn the basics of aseptic technique. We also have many articles written, and Simon has his blogs that you can just go in there and ask questions and get all the information you need from. So I would say definitely use those modes of communication as an added resource when you're trying to train your technicians in aseptic technique and personal protective equipment. So some of the pieces that you will need are alcohol that's greater than or equal to 70% isopropanol. You need eyewear so you don't spray yourself in the face with the compressed air. It's very important. A mask so you don't cross-contaminate. Gloves, and a lab coat. And a lab coat is more about making sure that you don't shed your microbiological organisms onto the plate. And the gloves are for the same purpose. So let's say you do get indicator organisms positive in your compressor air. The next thing that you have to do is you need confirmatory analysis. So it's not enough to just enumerate the microorganisms that are in your compressed air. You need to be able to say, well, I need to make sure that this organism that tells me I'm 1 CFU is actually E. coli. And so in order to do that, you need a third-party laboratory to do phenotypic or genotypic analysis of the organism. And that requires some specialty technique. And so usually a third-party lab does that. In the case here at Trace Analytics, we use phenotypic analysis, and it challenges the organism to essentially a 95 question survey about what sugars and foods it metabolizes. And based on those 95 answers, we get a molecular fingerprint, right? A metabolic fingerprint of the organism. And it'll say, based on these 95 questions or challenges, this organism has been deemed E. coli. And we can say with assurance to your customer or to you, hey, we know for assurance you have E. coli in your compressed air. And the next thing is the option of doing genotypic analysis. So the first one is phenotypic, and that is challenging the organism about what it metabolizes and how it acts. PCR is another way to challenge the organism, but this time you're asking it, what types of genes does it have that is specific to this organism? And are those genes present in the unknown sample that I have? Those are considered confirmatory analyses. These are not the only ones, but these are the most common. And so the next question that always pops up, and I want to kind of stay here for a little bit, is the presence of microorganisms in compressed air is asking you to go investigate your filtration or your pipeline functionality. And what I mean by that is oftentimes you guys get stopped right at having microorganisms in your compressed air and you don't know what to do. And so I wanted to take a little bit of time here and talk about that. So when you have microorganisms in your compressed air and particulates and water and oil, you need to start asking, let's look at a out of spec investigation, right? Let's start there. Let's do an investigation on the filters and are they functioning and are they working? And so you have pre-filters, you have coalescing filters, you have carbon filters. And based on what you need your point of use air for, you need to be choosing the most appropriate filter. And so if you are trying to get down to food grade air, you're obviously not going to just have a pre-filter that only goes down to class 3 air. That's just not going to cut it. You may need to have a higher quality filter, such as a coalescing filter that can reach a class 1 or a carbon filter that can reach class 1. A lot of these protection levels are for breathing air, medical air, food industry when it's in direct contact and not used for pneumatics, when compressed air is being used as an ingredient. So keep in mind to always, if you have microorganisms in your air, to go start at the filters and work your way back. That is one place to start thinking about. The other one is the pipeline functionality. You want to be thinking about, okay, you've heard that oil-free compressors are all the rage, and you want to get into oil-free compressors. So you convince your boss to buy you an oil-free air compressor. The problem is you've kept all the piping the same. So when you install your oil-free compressor on an oil compressor system, what you're doing is just changing out the compressor, but the air is still contaminated with oil from the previous compressor. So when you're thinking about validating a compressor for food grade air, you need to be thinking about the pipeline functionality. And is the piping appropriate for the compressor that you have? Is the piping appropriate for the compressed air grade that you want? You certainly wouldn't want to use black iron piping for food grade, right? You certainly wouldn't want to use copper or aluminum. These all fittings and tubings, they all shed particles. You want to be using the best materials for direct contact and ingredient used compressed air. And what that looks like is stainless steel or conductive polymers. When you do seals, you want to try to avoid any threaded or rubber O-ring compressions. You want to move towards stainless steel compressions. You want to move towards fully welded seals. and sealant types. And that's the same for sealant types as well. Welded is the best way to go, right? There is no risk of degradation there, because you will get degradation if you use putty or if you use PTFE tape. And same for the valves. You want to make sure that you're not using these rubber seals for food grade compressor usages or ball valves with rubber seals. You want to be using stainless steel shutoffs. You want to be using particle free stainless steel as well. So you want to be mindful of what it looks like to be using the best materials with the most efficient filters to get a very clear understanding of the functionality of your compressed air system when it comes to microorganisms. And so when we're thinking about what a report looks like, this is a question that I challenge you to ask yourselves. Is the analysis type that you're choosing adequate for your end product? So what I have here is it's a dummy report. It's an example of what a single ISO 8573-7 report would look like if you tested one point of use at your facility. So when we look here, you'll see you have the unique identification number, you have your controls, sterility blanks, and all of that. But also what you have is contamination under TBC, total bacterial count. And so while you're sitting there as a quality person who has requested this report, all you see is the 8 CFUs per cubic meter. What you're not going to see is this plate. You don't typically get images of plates from third-party laboratories. And that's because it's just not time efficient to provide that type of data. And so typically what laboratories do is they offer an additional package which will allow you to identify the colonies that are differentiated on your plates. So like you see here on the right, you have red colonies, orange colonies, yellow colonies, fuzzy colonies. So this is an actual customer provided compressed air sample. And so this is the level of differentiation that the laboratories see from compressed air. This is the level of variety that we see. When you guys get the reports, all you're seeing is an enumeration if you're choosing the basic package to provide. And so what I would challenge you is to, provide better quality for your customer, better quality for your employees by doing indicator organisms and identification. And so if you look at the report now, you'll see that it's changed and we've differentiated the colonies by their gram reaction. So let's say in this example, we have 4 gram-positive rods and 4 gram-negative rods. And from there, we did biolog analysis, and we found that we isolated E. coli and we isolated S. marcescens, right? And so those numbers next to the organisms is a quality value. It's a qual value. It's my confidence. The laboratory who saw this is 90%, 96% sure that we've isolated E. coli. Now, what this does is it allows you to provide more assurance, more control of your air, more control of your plant. And it allows you to troubleshoot more from that point of use. You know now that you may need to clean your piping. You may need to find the E. coli source. You may need to find the source of the gram-negative rod. And so that is invaluable information because now you can go to a filter manufacturer and say, hey, I have these gram-negative rods. They are microorganisms. They are contaminating my compressed air, and I need to find a way to get them out of my piping system. How can I do that? I need to find a way to get them out of my storage air. How can I do that? And they should be able to troubleshoot with you the ways to alleviate those types of issues. And so I would challenge you to ask those questions of your end product. Let's see. And so when I'm thinking about how Trace Analytics can partner with you, I think about it in terms of what we talked a little bit about, which is we're an accredited laboratory. We're 17025, which means we have to undergo rigorous quality compliant audits. We have impaction samplers for you guys to rent or to purchase. We handle all the training. We handle calibrations of the equipment. We can provide testings for particles, water, total oil, gases, environmental testing. All of this can be daisy chained with environmental testing. So all of this information that you get from your compressed air, you can also combine it with ambient air. And then you get an entire snapshot of the level of control in your food or beverage facility. And that is super invaluable. We are available Mondays through Fridays to do any customer service representation calls or to troubleshoot. So with that, I will take any questions that you may have. I wanted to leave a little extra time for us to talk because usually after my microbiology talks, I have a lot of questions. So I wanted to make sure that we had enough time to address all your questions. So please, let's hit your chat and I'll meet with Simon and we can start answering those.
Let's do it. Okay. Just to say, it is being recorded today, everybody, and we will follow up with an email to everybody a couple of hours after, and it will include the recording, a copy of the slide deck, and what else? Certificate. Yeah, get a certificate of attendance as well. Can't be bad. Okay, so let's start with the questions. Question from Jay. I have historically used the 2013 Parker-Ballston white paper, reducing contamination risk of compressed air in food plants. There are some updates needed as GFSI standards have been updated, but is this still a good white paper to support plant risk assessments?
Yes, 100%. I am a huge proponent of Parker-Ballston's white papers. They're a wonderful resource, and they have great plant risk assessments. Also, if you come into our AirCheck Lab website, we also have risk assessments tailored to specifically to 8573 questions for risk assessments in your food plant.
Okay, I've just copied and pasted that white paper title in the chat bar for anybody that wants it. Mohammed, is guideline from the British Compressed Air Society recognised? I'm talking about food and beverage grade compressed air practice guideline 102.
Yeah, so BCAS is the British Compressed Air Society. Does recognize many different guidelines. And what is good is that many of those guidelines are so vague for compressed air testing that really you can just lean on ISO 8573 as an umbrella and you will be in compliance.
Okay, sounds good. Another question from Mohammed. Market. Okay, we can also add terminal sterilizing filter at each point of use when sterility is mandatory. Can we also add that?
Absolutely. Yeah, absolutely. I think that's a wonderful addition. And we're starting to see more of that in the industry for food, beverage, and controlled environments. And that's the use of UVC for sterilization. It's the use of terminal sterilizing filters. It's the use of kill steps with compressed air. We're seeing all of that and the introduction of more dryers as well.
Okay, thank you. Question from Solomon. Brilliant analysis on compressed air in relation to microorganisms. However, Another risk analysis on compressed air is physiochemical contamination such as oil, metals, paint, etc. So how do we test for physical chemical contaminants such as paints, oil, metals, or non-metals in foods? Thank you.
Yeah, so what you're referring to is the rest of the contaminants. So today we focused on microorganisms, but there is a way to test for oil aerosols. There is a way to test for oil vapors, particulates, gaseous contaminants from paints. I'm talking about hydrocarbons. I'm talking about VOCs, volatile organic compounds, semi-volatile organic compounds. All of those, even metals, particulate metals, we can have those identified through a third-party lab that we use. All of that is captured inside of 8573, all of the testing methods. And at Trace, we provide a kit, one single kit that can do all of those particles, water, and oil tests. And then it's a separate kit for microorganisms, as I showed you before. But that's a really great question and something that I like hearing that people are thinking about.
Yeah, for sure. Another question from Mohammed. What is apparent rugosity required for stainless steel piping? I don't understand the question. You probably do.
Yeah, so we could shorten it and say, what is the requirement for stainless steel piping in a food grade usage? And the answer I would give to you is it is solely dependent on the risk assessment that you guys do. And so the first place to start is to go talk to your compressed air OEM. And once you go to the OEM, you will tell them, hey, I want my compressed air at this point of use to be 222 or to fall under GFSI or to be in line with BCAS. And I want them to provide me the piping that is appropriate and the filtration that's appropriate to meet those classes. And that's how you go about doing that.
Okay. Next one from Mohammed again. What kind of air sampler do we need for compressed air?
So, and the specific air sampler that you need is an impaction sampler. And so what impaction sampler is when the point of use air is slammed onto a nutrient auger. And that is an impaction sampler. But as I mentioned earlier before, not all impaction samplers are the same. So there is a small mobile impaction sampler that's available on the market today that is not appropriate for enumeration or quantification. It is only appropriate for qualification of your air. So basically saying it can tell you if you have microorganisms or not. It cannot tell you information regarding how many microorganisms you have. And so if that's important to you, then you need to be mindful. Plus, if you are held to ISO 857 compliance, then you have to use an impaction sampler that is compliant to quantifying the final microbial results.
Okay, got it. Question from Trang. What are the acceptable limits of microbiological results?
So right now for compressed air, at least in 8573, there are no limits. If we go into pharma, which is a little outside food safety, but it's more of a controlled environment, many of our food and beverage customers actually lean on pharma for some of their requirements, and they have particulate requirements on that end, and some microbiological limits as well. But I will say for pharma in a controlled environment, they are at less than 1 or less than 10, depending on the final usage. And those numbers are CFU, colony forming units, per cubic meter. So that's less than 1 CFU or less than 10 CFUs per cubic meter is typically what we see in a very controlled pharma environment that many of our food and beverage customers use.
Okay. Question from Rayanne. Is the bacteria in the air or on the piping system? How did you sample to get E. coli from air? What is the procedure? Thanks.
So that is a good question. And so when you are sampling your compressed air, the air is so pressurized and is coming out at supersonic speeds. The air is not on the microorganism is not attached to the piping system. So it is in the air. So that's your first question. Second question is, how did you get E. coli from the air? Well, that happens if you have an inlet that is not in a very controlled environment or that is not in a clean environment. You'll remember there was a story a few years ago where the inlet was in a basement that was at the very, very bottom of a chimney stack. And so a bird had nested at the very top of the chimney stack and was defecating. And so all that waste was being taken in to the inlet of the compressor. And if their filtration failed, and it did, they ended up getting contamination from that environmental contaminant. So that is a way that you can have E. coli, or you can have these indicator organisms in your compressed air. And again, it starts with investigating the at times aftermarket filters, aftermarket dryers of your compressor systems and your piping.
Okay. All right. Question from Nigel. Do you have a partner lab in the UK where we can hire the impactors from and have the analysis carried out?
So we don't currently have a partner in the United Kingdom. If all you needed was an impaction sampler, we might be able to purchase you one and we can hold it for you, or you can purchase one and we can definitely send it to you. We can validate it here in-house, keep all the parts of it calibrated for you. That is definitely an option, but we do not have a partner lab in the United Kingdom.
Okay. Opportunity there for somebody.
Yeah.
Edwin, we do not have compressed air. Is the testing of ambient air, say microbial air density, also governed by ISO 8573?
So no, for compressed air, that 8573 is strictly compressed air. ISO 14698 is ambient air, compressed air, surfaces, environmental, clothing. That is ISO 14698. And that is what governs the controlled ambient air.
Okay. Anna, question from Anna. What are the acceptable limits of microbiological results for food contact?
Yeah, it's really frustrating because this question pops up every single time I have a microbiology webinar. And it's unfortunate that my answer has to be the same every time. And that is, it's strictly risk-based. What is acceptable to one's ingredient plant will not be acceptable to somebody else's ingredient plant. And so what we need to do is get more people on the regulatory standards boards to start pushing legislation to get limits so that we can unify this. Again, I did mention earlier, Anna, that we have many food and beverage customers that lean on pharma that do have regulations and do have limits for microbiological contamination. And for them, it's less than 1 CFU and less than 10 CFU per cubic meter.
Okay. Long question coming up from Alan. I work in an edible oil business where we have to discharge the oil out of the shipping tank using compressed air. There is a HEPA filter fitted to the compressor to trap any dust particle, airborne contaminants, etc. The data sheet for the filter didn't state a micron size, just a percentage efficiency rating. In a recent BRC audit, so I couldn't provide this info. Is this absolutely necessary to have the filter pore size?
Yeah, definitely. You want the filter pore size, and that's because microorganisms are tiny. So if you end up getting a HEPA filter that's only good enough to filter out pollen, but it's not good enough to filter out microorganisms and viruses, then you might have a serious issue. But again, I would recommend a risk assessment, and I would recommend getting that information.
Okay. Question from Karen. How long should be the time exposure for compressed air monitoring?
So typically what we do is you need to capture 1 cubic meter of air. 1 cubic meter is 1,000 liters of air. So many of these impaction samplers are limited by the PSI that can be exposed to them. And you definitely do not want to break the auger with certain velocities of air, flow rates. So we recommend 100 liters per minute for 10 minutes, and that is the standard.
Okay. Question from Prem. There are various filtration methods, so which are the best methods of filtration and filtration module?
Yeah, so the best filtration and filtration modules would be analyzing based on your risk assessment whether or not your point of use is acceptable for the filter that you use. So if you want food grade air for a clean room, you're probably going to want class 2 or class 1 for particles. And so you need to make sure you have a coalescing filter or a carbon filter that can go down to class 1 or class 2.
Okay. A question from Saida. Which standard to follow to check compressed air?
That's ISO 8573.
Yeah. Patricia, can ambient air have fungal growth?
Yes, 100%. We have seen black mold. We have seen your just run-of-the-mill slime molds. So yes, 100% it can have mold growth. And again, if you see it, you need to start looking at your filtration.
Right. Got it. Araceli was asking, what about Mexico? Do your services cover Mexico?
Yes, 100%. We do support Mexico and Canada.
Okay, so get in touch. Contact details will be on the slide. Jonathan, is it very possible to achieve 100% compressed air with no trace of contamination?
With no trace of microbial contamination, yes. You will likely have a pressure dew point, so you will have so that's a tough answer. Microbial contamination, yes, it is very possible and achievable to have 0 microbial growth in your compressed air. But you'll only know that if you test. It's not something that you can have assurance of if you're not testing your compressed air. For particles, water, and oil as well, it's not about having no trace of contaminants. It's about being inside the limits of the purity class you want.
Okay. I think you've disappeared off the webcam for some reason, for me anyway. Mohammed, is ISO 8573-7 mention slit sampler? Is it the same technique as impaction sampler? Slit sampler and impaction sampler.
So a slit sampler is a type of impaction sampler, so it is appropriate.
Okay. And Karina, if there are indicator organisms in the air and the compressor was recently used and contacted food product, after risk assessment, there is doubt of the integrity of the food product. Would this trigger a recall?
It's possible.
Mm,
It would just depend on your quality protocol.
yeah. Preventive action is the best method. Yes. Um, not guess, it is. Jonathan, is there a fast way of testing contaminant in air for urgent results?
Hey guys, I thought I lost y'all for a second. Sorry.
Yeah, you're there anyway. It doesn't matter. You've gone again, but it's fine. There. Jonathan, question. Is there a fast way of testing contaminants in air for urgent results? How quick can we turn them around?
Yeah, so what we typically do for urgent results is two different methods. One, we give you preliminary results like every 2 days, and we provide an unaccredited preliminary report of your microbial growth so that every 2 days you know, okay, I still don't have growth, we're okay. Or, oh my God, it's been day 2, we already have a bunch of growth. That's one way, and that is because ISO 8573 requires a minimum of 10-day incubation. You can't rush the minimum of a standard. But what we can do is keep you updated with preliminary reports. The second thing we can do is do custom incubation periods. So we can so we can no longer test to ISO 8573. What we do is we create a specification for you and we incubate for 2 days and 5 days or whatever your risk assessment has deemed an appropriate incubation period. And then we give you that information. If you feel confident with that, then we can move to ISO 8573 with more assurance that you have control of your system. That is another way. We can rush ship kits, Pinocchios, we can rush ship plates, anything like that. And the turnaround can be inside of 24 hours for the report anytime that you need it.
Okay, good. That one. A question from Helen. Is using compressed air to clean tooling to seal products and to clean food contact belts considered food contact grade air?
Is using compressed air to clean tooling to seal products. It's considered indirect air. So you are using the compressed air in your facility, but it is indirectly going to be in touch with your product. So that's considered indirect. But it is considered food grade air. It's just indirect usage.
Yeah. Got it. Patricia, kindly advise the recommended agar to use for fungal growth monitoring.
So if you want to do fungal growth, what you need to do is inhibit the growth of the bacteria so that you get a real image of the fungal community in your air. And to do that, you would use MEA media, malt extract agarose. You could also use SDA, which is Sabaron dextrose agar. Both are selective medias that inhibit the growth of bacteria and feed the molds and fungi.
Okay, got it. Question from Madhu Kumar. What's the level of risk when using compressed air to blow packing materials, well, we just covered that, haven't we? Where you're using air on surfaces. So I think that's covered. If not, feel free to ask another question. Alyssa, can a settle plate be useful in compressed air testing? Based on your training today, the impaction sampler seemed to be a must for testing, correct? I have not seen a specific method dictated by SQF.
Yeah, that's 100%. So I have an article that I think we wrote for IFSQN or maybe, but anyway, You cannot use a settle plate for compressed air testing. You cannot do it. And why is that? Because you will be mixing ambient air with compressed air, and you will not know which of those variables caused you growth. So you have to use an impaction sampler that is a closed piece of equipment to test compressed air. End of story. So SQF doesn't specify a method. What they specify is that you need to test your compressed air. If you go to ISO 8573, that is the explanation, internationally accepted by the facilities worldwide for testing compressed air. So use ISO 8573.
Okay. Question from Mohammed. Recommend what is the monitoring frequency for compressed air? I think that's the question. What is the recommended?
Yeah, so the monitoring frequency for compressed air, in my opinion, should be at minimum quarterly. And why is that? We have seasons, and even if your compressor is located indoors, your HVAC, all of that, everything is being subjected to the seasons, the humidity, the environment changing. So I recommend doing every 3 months of compressed air testing minimum.
Okay.
You can also do a risk assessment, which you would end up doing, let's say you have 100 points of use. You could do 25 points of use one quarter, another 25 another quarter, another 25 another quarter. It just all depends on the risk assessment that you do.
Okay, thanks. Karen, if I want to know the air quality in compressed air, how long should be the exposure time on the plate? Did we answer that before?
Yeah, so that's based on your impaction sampler, and typically it's 10 minutes at 100 liters per minute to capture 1,000 liters of air.
Okay. Another question from Patricia. When sampling compressed air, what are the likely microorganisms to watch out for?
So typically, if you get gram-positive organisms that are gram-positive cocci, which is typically found on your hands, you know that you have some aseptic technique issues and you need to undergo aseptic technique training. If you're finding gram-negative rods, you have serious breakdown of filtration or a serious breakdown of moisture in your compressed air, and you need to get that looked at. Gram-positive rods are sometimes spore forming, and so you need to be really mindful of your filtration at that point. So those are a little bit of the indications of organisms that you should be concerned with.
Super. Right. We've exhausted all of the questions.
Awesome.
And it's 1 minute. In fact, it's probably about 30 seconds spare. Absolutely brilliant. Excellent presentation as usual, Maria. Lots of great content and just like perfect answers. Amazing. Fantastic.
Thank you so much, Simon.
Yeah. So, yeah, thanks very much, Maria. Enjoy the rest of your day and enjoy your weekend.
Thank you so much. Bye-bye, y'all.
Take care. Okay, ladies and gents, that was brilliant. Maria Sandoval from Trace Analytics. Like I say, we'll follow up with the recording, the slides, certificate, and it'll have Trace Analytics contact details on there. They're always open to an email. They've got great resource on the website. If you want to send them an email, if you have a question that comes to mind, they'll answer it. And obviously, they've got lots of product services that can help you with your compressed air testing and food safety. Okay, so I'm going to put your certificate in the sidebar now if you want it now. Remember, you've got to add your own name to it. You can print and sign it or open it in image editing software and type it. It's pretty easy. Saves me a job of doing that for 1,100 people registered today. So thanks everybody for registering, and for those of you joined live and who will watch this recording, and we'll see you on the next one. Have a fantastic weekend. Take care, everyone.